Welcome to the Defense and Aerospace Report. I'm Vago Muradian here at the Gaylord National Convention Center outside Washington, D.C., where we're covering the Defense Advanced Research Project Agency's 60th anniversary conference and trade show, the 60th birthday of one of the world's most incredible technology organizations. And we're here on the exhibit floor to talk to Dr. Renee Wegrzyn, who is with the Biological Technologies Office. You do gene splicing work, and we're next to hundreds of the deadliest animals on the planet, your favorite and mine, mosquitoes. Renee, tell us a little bit about the gene work you're doing, because it's pretty incredible stuff. And what is the military and broader sort of societal application of it?
Great, thank you very much for the opportunity to talk with you. We're showcasing here the Safe Genes program. So I have a long-standing interest in genome editing technologies, and these are very powerful technologies that allow us to attach to DNA and alter sequences of DNA. And why that's really important is you could use that, for example, to cure inherited genetic disorders, but we're interested here in exploring other opportunities. How can we potentially look at disease vectors like mosquitoes, as we have here, and alter those mosquitoes' genomes so that they can be resistant to malaria or Zika virus, or that we could even suppress those mosquito populations to reduce disease incidence. That's a long-term visionary goal, but what the practical work that we're doing here at DARPA, we're not planning on any open release, but we want to establish the ground truth in the laboratory of what's possible with these technologies, how do they really function and work, where do they fail, and how can we mitigate the risks, any risks that they might present. So key to the Safe Genes program is not only establishing the ability to edit these genomes, but the ability to control, counter, and reverse the effects of genome editing.
Which is interesting, and I want to come back on. Well, so how do you reverse the process, right? I mean, so everybody is very, very concerned. I use the very bad example, right? But I mean, sort of the Jurassic Park, right? The altered animal gets into the wild and becomes an issue, but we've seen that with crop management, for example, where it's caused certain problems and displaced crops. When you and I were talking before the interview, you were talking about, you don't want to so change a species, for example, that one dies off and is replaced by potentially a worse species. But talk to us a little bit about the reversibility of this, and how do you engineer that? Because there's this sense that once you do gene editing, it's an irreversible process.
Right.
But you're looking at it as actually being a reversible process. Talk to us about how you do that.
That's right. So in the same way that you can think a disease gene previously was thought to be irreversible, now we have a technology to restore a healthy gene. In the same way, we want to see if there are technologies we can develop that if there should be a gene edit that's made that has an unexpected consequence, that we can restore that system. So you can use that by having a follow-on genome editing technology, or you can also start to develop countermeasures. For example, even small molecules where a gene editor would be dependent on the presence of that small molecule in order to spread. So if you wanted to keep that spread of, let's say, an engineered mosquito local, you could have pre-treated an area, for example, that would be dependent on a given molecule. You could also engineer in fitness deficits where it can't outcompete the wild populations. And so these are all hypotheses that we have, that whole broad range are things that we're really testing here in the laboratories to see if we can achieve those, which ones look to be the most promising technologies.
And these are all male, right? So they're non-stinging, and apparently people have had the courage to put their hands in there, which I think is lovely. And they're very hardy, aren't they?
Yes, they are. So these, each of these cages here has 2,000 male mosquitoes. These are wild-type, non-engineered mosquitoes. But they just kind of want to show you the example of...
They're the Whole Foods, friendly, all-natural mosquito, and I'm sorry.
Well, so this is the scale that we're doing our experiments on. So you can have a whole population, and you can look at the impact. How quickly does a engineered gene spread in this type of environment? What are the impacts? Can we really reduce the spread and incidence of viruses in the guts of these organisms? So when the females take a blood meal, that's where you have the threat of zoonosis or transmission. So can we stop that at that level before it really becomes a problem for the human?
One of the things you mentioned was that with genetic engineering, you can sort of tailor, for want of a better word, a release in a time and place where you need it. But that the genetic engineering would also allow the modified organism to actually die off depending on a whole variety of environmental factors. Talk to us a little bit, because I mean that's just frankly incredible, right? Because you're basically programming the animal almost as you would whether a munition or software. Talk to us a little bit about how you guys managed to achieve that degree of precision with something which is an incredibly complicated organism.
Right, so but what the program is doing is we're trying to test our hypotheses in what we would call a self-limiting manner. So we don't want these to go in perpetuity. So we've developed computer models that help us design different types of approaches to engineer the genes that would limit the spread to maybe only 5 generations, 10 generations before they're expected to die out, and then wild type populations can be restored. And so when we build those models, we then actually build those genetically as well and try to test them. One of the challenges with the mosquito is that it has a life cycle of about 2 weeks, which sounds very short, but in the laboratory and DARPA timelines, that's actually a long time. So we also have research in more rapidly reproducing organisms like yeast and small nematodes, where we can have much longer evolutionary timescales to see, how long do these persist? Can we really limit the spread of these organisms? And if they fail, how can we, mitigate against that risk going forward?
How? You mentioned it's the deadliest animal in the world, and it truly is. How many people die of mosquito-borne illnesses on an annual basis?
So on an annual basis, for malaria in particular, there's about 700,000 deaths. And so I don't know the statistics for other diseases, but add to that Zika and malaria and dengue and other diseases, it's quite significant. It's a global problem. Wherever a mosquito lives, there's mosquito-borne diseases.
And that's one of the reasons why the Gates Foundation is taking such a serious interest in this work.
That's right. Yes. So there are, as not surprisingly, there's many other investors that are really excited about this type of work and are also exploring this space that, we certainly hope we can contribute to that research field as well and give them alternatives to some of the other approaches. So what SafeGenes is pursuing is different than some of the other approaches, and a priori we don't know which is best, but we want to have our skin in the game too.
How scalable is this from the mosquito level to the human level, for example? I mean, what are sort of the connections? What do you learn, for example, doing this on a mosquito that is also applicable to humans,
right.
ultimately?
So, actually, what we're not showing you here today is we're really focused on the part of our portfolio for SafeGenes that's focused on mosquito gene drives, but we also have investments as part of this program looking at therapeutic applications. And so if we're really excited about using these tools to reverse the effects of disease, we have to also have a safety by design principle going forward that if there should be an unintended consequence, we can do something about it. One example I'd like to use is if you have an illness and you have to take a medicine, if you have an unexpected side effect that appears, you just stop taking that medicine and you can restore your system to normal. But if you're permanently altering your DNA and there's an unexpected side effect, that's going to continue to alter your DNA. And so I think it would be a great idea that we, in parallel, develop countermeasures that we can immediately halt that progression of introducing potentially off-target effects, or even reverse those effects if we want to move forward in a big way with therapeutics, which I think we see is probably going to be the future.
But if you also have another society who maybe has a different attitude toward weaponization of DNA, you would also be able to countermeasure that potentially in the event that, because that's a concern, right? Obviously,
yeah.
That there are folks who may not have the same standards or not have the same kind of approach, which is then very, very problematic for the person who, or the people that it's used against.
Right, that's right. So what was very important for me in establishing the SafeGenes program was that we have to explore the beneficent uses of these technologies because they're so powerful, we can't ignore them, but at the same time acknowledge that there's a potential for misuse, whether it's accidental or nefarious misuse. And if we develop the solutions to control and counter and reverse those effects, we're actually kind of solving for both. We're solving for the nefarious misuse as well as the accidental misuse. So that's a very big part of this program.
Absolutely fascinating. Dr. Renee Wegrzyn from the Biological Technologies Office at DARPA. Absolutely fascinating. Thank you very much, and best of luck with the program.
Thank you very much. Take care. Thank you.